All right, can everybody hear me? Okay, great. So thanks, Dan, very much for that very nice introduction. And as I kind of alluded to in my early morning talk, hopefully you won't hold it against me that I'm a Los Angeles Dodgers fan. So sorry, Rockies, but you guys get another chance tonight, I think, against the Cubs. But I'll be rooting for the Cubs, sadly, because my son is actually a Cubs fan. So anyway, got that out of the way. My talk is, as Dan kind of alluded to, I'm focused on myeloproliferative diseases, and I'm going to tell you about a few different things that we've been working on. So just to give you an overview, just first I'll briefly cover some background on the role of aberrant signaling in the MPNs. I'll tell you about a story that we published in Leukemia about a year and a half ago or so, where we identified NFKB hyperactivation in myelofibrosis and secondary AML. And then recent work, which is not published yet, where we have begun to focus on the interplay between inflammatory cytokines and the dysregulated signaling pathways in these diseases. And then, fortunately, we've been able to translate this to the clinic, where we are specifically targeting NFKB hyperactivation in MPNs. And then lastly, assuming I have time, I'll tell you a little bit about a separate story in polycythemia vera. So, just some basic background on the MPNs. So these diseases, the three main subtypes, polycythemia vera, essential thrombocythemia, and myelofibrosis, share a number of features, and yet they also are distinct. So for instance, polycythemia vera, the classic hallmark, of course, is too many red blood cells, elevated hemoglobin. Essential thrombocythemia, too many platelets. Both of these diseases carry with them an inherent risk for thrombosis, both arterial and venous. And over time, both of these diseases can evolve to myelofibrosis. So myelofibrosis can occur post-PV or post-ET, but also can occur de novo as primary myelofibrosis. And some of the typical features, of course, underlying bone marrow fibrosis, constitutional symptoms. So patients with MF tend to have more severe symptoms in general than patients with PV and ET. Extramedullary hematopoiesis, and in some cases, quite massive splenomegaly. Now, I'm going to focus a large part of my talk on inflammatory cytokines, which are known to be abnormal across the MPNs, but if anything, most severe in the case of myelofibrosis. MF can transform to secondary AML, and when that happens, the prognosis is dismal, even worse than it is for de novo AML. And in some cases, PV or ET can directly transform to AML. So these diseases are clonal disorders, and they're thought to originate in some hematopoietic stem or progenitor cell. And you have mutations such as the JAK2 V617F mutation or other driver mutations such as this MIPL mutation or a mutation in CALR, calreticulin, which, again, are thought to arise in some primitive progenitor cell, and then their effects are transmitted throughout the entire myeloid lineage. And so this results in one of these three MPN subtypes. And again, myelofibrosis with these specific features can transform to AML. So this is much of the work in my group is focused on myelofibrosis because it has the worst outcome besides secondary AML. And all of these three MPN subtypes share this characteristic hallmark of JAK-STAT activation. So classically, most commonly, the JAK2 V617F mutation. It's an activating mutation in the JAK2 tyrosine kinase, which results in either constitutive activation of JAK2 or hypersensitivity to cytokine stimulation. Normally, you need these cytokines such as TPO or GCSF to stimulate downstream signaling. And in a sort of simplistic sense, this results in hyperproliferation, such as what you see, for instance, in polycythemia vera with the elevated hemoglobin. Now, with the JAK2 mutation being identified in 2005, it was an obvious target for therapeutic intervention. And so ruxolitinib is one of several different inhibitors of JAK2 that went into clinical development. And so what we have found clinically with using these drugs such as ruxolitinib is that ruxolitinib and the other JAK inhibitors can pretty reliably result in improvement in spleen size. So this is a waterfall plot showing each individual patient as a line, and then these are all negative because there's a shrinkage in the size of the spleen, and that's just not, it's not just a number. These patients have improvement in their overall symptoms because of that, and that's also shown here in this what we call total symptom score, which improves dramatically with ruxolitinib, and then in this study compared with placebo, as you would expect, no significant improvement. So this alone is really meaningful clinically for patients. I have patients who, they've lost 30 pounds, they come back, they've gained it all back, and then they actually start complaining because they didn't want to gain all that weight back. So, it's really quite dramatic in some cases. And yet, there are limitations to these drugs. So, these drugs, such as ruxolitinib, they do not appear to be capable of eradicating the underlying malignant clone. So what I mean by that is if you measure the JAK2 mutant allele burden, let's say it's 80% before you start treatment, it might go down a little bit. So let's say it goes down to 60% or 50%. It will not go down to zero. It's not like in chronic myeloid leukemia where with BCR-ABL we have targeted inhibitors and we can reliably get the level down to microscopic levels, 0.01% or even undetectable by very highly sensitive measures. That is not the case for these diseases, and that raises the obvious question of why. And so we have been focusing on this question for a number of years in my group, and really by thinking of it this way, which is that this cartoon I showed you is obviously very simplified. And what if we think of it more like this, okay? So, and this is even just a cartoon that's oversimplified, but it shows you that there are a number of different, classical signaling pathways, and these can intersect in different ways, both through crosstalk from JAK2, as well as through, for instance, upstream cytokines such as TNF-alpha, which can activate, for instance, NFKB, which of course I'll tell you more about as we go forward. And then you also have to consider that if you introduce a perturbation to the system, such as a JAK inhibitor, what does that do? So let's just say, for instance, one of these collateral signaling pathways is not really that fundamental to the disease in its initiation, but when you inhibit JAK2, maybe these other pathways become recruited or become more important, so that you have to consider or potentially target those pathways as well. So in studying this kind of thing, as I'm sure most of you are aware, it's very difficult to use quote-unquote traditional biochemical approaches. You're not going to do a Western blot on 20 signaling proteins in primary cells, in stem and progenitor cells from patients where cell number is limiting. That's just not going to be feasible. So to address this, we turn to mass cytometry, or cyto. And in a nutshell, this is essentially a hybrid of flow cytometry and mass spec. So you have here, for instance, with traditional flow cytometry, your antibodies are conjugated to fluorophores, and as you get to add more and more colors to your experiment, you have a problem where these fluorophores begin to overlap, and so that becomes limiting. Now, with mass cytometry, we use the same antibodies that you use for flow, but they're conjugated to heavy metal tags, and the readout is mass spec. And now you have a much wider spectrum that's available. The channels are very clean, and so there's very little issue with signal overlap. And with this specific instrument called the cyto, you can still retain single cell resolution. So that's key. So now you can still look at individual cells like you can with flow, but you can measure many more parameters. So it's very common now with this technology, and I know you guys have cyto here, so some of you have used this. You can easily get into the 40 parameter range with single cell resolution. And so for our purposes, this is really powerful because we want to be able to look at these different signaling pathways, and I've outlined here in white all the different markers for which they're good validated antibodies to be able to use via mass cytometry. And so this allows understanding of intracellular signaling in these diseases. So also keep in mind that we're not just interested in one cell type. So we are very interested in the stem or progenitor cells, but also in these mature cells. And I'll show you as we go forward why we've become in particular interested in, for instance, monocytes and their role in cytokine secretion. So we want to be able to look at all these different cell types and how they relate to each other in terms of their different phenotypes. So starting with this kind of approach, a number of years ago, we just said, okay, let's survey a number of intracellular signaling molecules in different patients with MPNs. And so this is one of the first experiments we did, and focusing on CD34 positive progenitor cells. And then I'm showing you 3 normal controls, 4 patients with myelofibrosis and 5 with secondary AML. And this heat map shows the relative levels of these different intracellular signaling proteins, and essentially yellow is high, blue is low. And you can see right off the bat that there is considerable variability from patient to patient. It could in part be patient as patient variability. It can in part be somewhat technical related to sample variability, et cetera. But there are themes that emerge, which is, of course, not surprisingly, you see evidence of JAK-STAT activation. But perhaps more surprisingly at the time, we see evidence of a lot of other signaling pathways being dysregulated. And the one that caught our particular attention at the time was NF-κB. We were at the time measuring IκB alpha, which is a negative regulator of NF-κB signaling. So we weren't really sure what to make of this initially because you see these elevated basal levels, and does that mean that NF-κB signaling is actually downregulated or is it hyperactivated? And so, and just to illustrate that point here, you can imagine that NF-κB can get activated in a number of ways. So for instance, JAK-STAT signaling could crosstalk directly with NF-κB. That's known to occur. JAK-STAT signaling can lead to upregulation of TNF-alpha production, and that can then act on a neighboring cell in a non-cell autonomous fashion and activate NF-κB that way. So we weren't really sure what was going on, although our hypothesis was that higher levels of IkB alpha actually reflected chronic hyperstimulation of NF-κB, perhaps through TNF-alpha. And so we then added into our panel an antibody for phosphorylated P65 rel-A. And that's a more direct measure of NF-κB activation. And then here what we found was that in patients with myelofibrosis, the basal levels of phospho-P65 were higher than they were in the normal controls. And that was true in the basal setting. And in some cases, if you stimulated with TNF-alpha, you would get an even hypersensitive response. So both basal hyperactivation of NF-κB and intrinsic hypersensitivity to cytokine stimulation. And this is just a summary of the experiments we did to evaluate that, showing that although, again, there's variability, overall we do see evidence of NF-κB activation frequently in patients with myelofibrosis and secondary AML. So that was all by Cytov. We wanted to turn to another data set to try to validate this. So here we looked at gene expression data. This was a published gene expression data set from CD34 cells from patients with myelofibrosis. And when we did gene set enrichment analysis, we found that there was an enrichment of NF-κB dependent gene expression in patients with myelofibrosis, both JAK2 mutant and JAK2 wild type, although perhaps a little bit more significantly in JAK2 mutant patients. And then we looked back at our Cytov data again and asked ourselves, well, what is the relationship between JAK-STAT activation and NF-κB activation? So here, first I'm just showing you a very simple correlation analysis between phosphorylated STAT5 and phosphorylated P65. And you can see there's a rough correlation. It's really not that strong. But we wanted to look at this in a more sophisticated manner, so we looked at it at the single cell level. And so here, using an algorithm called DREMI, which looks at each individual cell and, again, the correlation between two different markers, here again, STAT5 versus phospho-P65, what we could see is that there was a relationship between the two markers at the single cell level, and in particular, even more so in the patients compared to normal. So this suggests that the pathways are linked, and perhaps even more specifically in the patients compared to the normal. Okay, now we wanted to then ask, well, what is the impact of ruxolitinib? And so we did that. We looked at this is in vitro, before and after exposure to ruxolitinib. And what we found is that the levels of phospho-P65 did not significantly decrease. So the NF-κB hyperactivation was insensitive to JAK2 inhibition. And this was both at MF and SAML. And then this was what I just showed you was an hour-long exposure to the ruxolitinib. So we wanted to ask, well, what happens with a longer-term exposure? Do you just need more time to be exposed to the JAK2 inhibition? And the answer is no, that does not rectify the NF-κB activation. In fact, what we found, if you look at, for instance, this is a patient with myelofibrosis, and this is specifically gated on the CD34 cells. In the basal setting, which is no stimulation, no inhibitor, just by virtue of being in culture for longer periods of time, the phospho-P65 levels go up and up. And in the presence of ruxolitinib for the same amounts of time, it does not fully resolve that. So our idea with this is that these cells are spewing out cytokines such as TNF, and the ruxolitinib cannot overcome that. All right, so looking at it another way. So everything I just showed you is what we published in our paper in Leukemia in 2017, and everything going forward is unpublished. So and this is another way of looking at the effect of ruxolitinib. So these are Visney plots where every individual cell is plotted as a dot, and then you can color code it for whatever marker you want to look at. And so, for instance, here is just giving you the landscape. Here's a normal control versus a patient with myelofibrosis pre and then on treatment with ruxolitinib. And so again, this is just showing, for instance, CD34 highlighting the stem and progenitor cells, CD14 highlighting monocytes, et cetera, CD3 highlighting T cells. So these same plots are then shown on the right here, where we now have color-coded this for phospho-P65. And again, here's the normal control versus the patient with myelofibrosis pre and then on treatment with ruxolitinib. So this is actual therapeutic treatment with ruxolitinib. And what you can see is that the phospho-P65 levels are higher in the patient, and they're not just high in the CD34 cells, they're also abnormally increased in the T cells compared to the control. So this is pretty strong evidence that, at least in the case of the T cells, NFKB is getting activated in a non-cell autonomous fashion, because we know, both from the literature and from selected cases where we've confirmed it, that the T cells, by and large, do not harbor JAK2 V617F. So they cannot be directly activating NFKB that way. So this is a widespread non-cell autonomous phenomenon, and that upon treatment with ruxolitinib, these levels do not get normalized. So both in vitro and in vivo, we have evidence that NFKB activation is insensitive to ruxolitinib. Okay, so this is just to remind me and you again about the different ways that this could be happening. So, TNF being the prime suspect for how this is occurring. So we looked at plasma levels of TNF, and similar to published data, we confirmed that compared to normal controls, the plasma levels of TNF are significantly elevated. And then we had this cohort of patients who also received treatment with ruxolitinib. So we looked at pre and then post or on treatment with ruxolitinib. And although the levels go down a little bit, they remain significantly abnormally high compared to the normal. So I think this is something where there was literature out there from the clinical trials with ruxolitinib showing decrease in a wide array of inflammatory cytokines in patients treated with ruxolitinib. That is true. However, what was not clear from those papers is that it still remains abnormally high. So ruxolitinib does not effectively normalize inflammatory cytokine production. This is what we have found from our studies. And here is showing you an array of different cytokines where we looked at normal controls compared to MF patients pre and then post ruxolitinib and in secondary AML. And the theme here is that many of these are significantly elevated, and while some of them do decrease to some extent with ruxolitinib, they do not go back down to normal. So clearly, ruxolitinib is not capable of completely normalizing this inflammatory cytokine production. So one obvious question then is, well, what are the cells that are producing these cytokines? And I really thought this was an important question to go after, because the plasma cytokines just give you this, kind of net level of the cytokines. It doesn't tell you where they're coming from. It doesn't tell you, that maybe specific cell populations are the ones that are really the bad actors here, and if you could target those, perhaps that would be effective therapeutically. And so we wanted to get at this, and sort of conveniently, CyTOF was a way that we could do that by essentially using the same approaches we use for signaling, but now focusing on cytokines. So we simply swapped out our signaling markers and added in a bunch of cytokine markers. And so when we did this, one thing that became quite clear is that the specific cytokines, where they're coming from, is variable. So while many of them are predominantly produced by monocytes, certain cytokines are produced by different types of cells. So here's a couple of examples. So TNF, primarily produced by monocytes. So again, in these visiting plots, you have CD14 highlighting the monocytes. And then TNF alpha, you can see here, is coming, in the case of the patient with myelofibrosis, down here, from the monocytes predominantly. You don't really see much going on in the normal because there just really isn't much basally in the normal setting. And another thing which you can probably see is that the monocyte population is different in the patients. So you can see that with the Visney algorithm, it's grouping the normal monocytes off to the side here, and that the patient monocytes extend to over here. So we haven't had a chance to really delve into that in detail, but I think it's a hint that these monocytes are not normal monocytes, of course, but they might have specific features that we can kind of dig deeper into with further analysis. So that's TNF, a good example of one of several cytokines that come predominantly from monocytes. Here's an example of IL-8 where, if anything, it's primarily produced by CD34 cells. So you can see the CD34 cells highlighted here, and this is where the IL-8 is predominantly coming from. It also comes from monocytes to some extent. And then IL-10, which is coming from both monocytes as well as NK cells. So you can see the NK cells highlighted here, and the IL-10 being produced there. So again, it depends on the specific cytokine. Majority are from monocytes, but there can be other sources as well. So this gives you a further idea. So these are heat maps showing several different progenitor and myeloid cell populations and the different cytokines, and then looking at, in this case, 2 patients with myelofibrosis who are JAK2 mutants. It's probably a little difficult to see all the details, but 2 patients here, next all normalized to the control, so that you can see the control column is black, and you can see a whole lot of yellow. So that tells you that there are a lot of cytokines that are abnormally being produced, and in multiple different cell populations. And then, albeit with a relatively small sample size, it seems that this is a universal theme that in patients who harbor the MIPL mutation or with calreticulin mutations, we see a similar phenomenon as well. So again, in myelofibrosis, this abnormal cytokine production is a hallmark that's shared across different genotypes. Okay, so, and again, the next obvious question is, what are the intracellular pathways that are driving this aberrant cytokine production in myelofibrosis? And so we then conduct experiments where we expose the cells to different stimuli to see what would accentuate cytokine production. And so here we looked at TLR ligands. And so here, for instance, is PAM3CSK4, and this is focusing specifically on monocytes, and we're looking at TNF. And so what we found, for instance, here with a normal control versus 2 patients with myelofibrosis, you can see that in the basal setting, there is a subtle but quite obvious increase in TNF positive cells in the patient versus the normal. Here, not so much. But when you stimulate with PAM3CSK4, you see a robust response in the patient cells that's not nearly to the same extent in the normal. So again, these cells seem to be hypersensitive to cytokine production in response to TLR ligand stimulation. And the same was true in this example with R848, another TLR ligand, in this case looking at IL-6. So you see a robust induction of IL-6 to a greater extent than you see with the normal, and again, this is specifically in monocytes. We did dose response curves with, in this example, with R848, and the blue line is normal, and then the 3 individual patients with myelofibrosis, again, specifically on monocytes, and this is looking at TNF, and you can see that at the lower concentrations of R848, we see this hypersensitive response. As you get to the higher concentrations, that's not necessarily the case, but that's not entirely surprising. You see this hypersensitivity to lower concentrations of R848. Now, in contrast, certain cytokines were not responsive to TLR ligand stimulation. So here's an example with TGF-beta, where the levels are flat. They are significantly increased in the patients compared to normal, but they don't really respond to TLR ligand exposure, and I'll come back to that point in just a couple more slides. So one of the other things that we looked at in terms of stimulating cytokine production was thrombopoietin, or TPO, and somewhat surprisingly, we found that monocytes responded to TPO stimulation. And I say surprisingly because you wouldn't normally think, or at least I wouldn't have thought, that monocytes would respond to TPO. This, MIPL expression is thought to generally be confined to progenitor cells or megakaryocytic cells. And so it was a little surprising that this was the case. So you can see, for instance, here we're looking at IL-8, and with stimulation with TPO, we can see that there's increased production of IL-8, and here with IL-10. And in this example here, also with IL-8 and TNF, you see the same thing, that TPO stimulation is resulting in cytokine production from what appear to be monocytes. I'm going to come back to that point as to how that might be occurring, but first I'll just show you that this cytokine production, again, was hypersensitive compared to normal. So in this case with TNF, you see in response to TPO, a much more robust induction of TNF production compared to the normal. And when you look at the effect of ruxolitinib, we found that this TPO induction was sensitive to ruxolitinib. That was not necessarily surprising, but perhaps more importantly in this case, we found that the basal increase in TNF positive cells compared to normal was not abrogated by ruxolitinib. So further evidence of the incomplete effect of ruxolitinib. Now, these TPO stimulation experiments in all the cytokine experiments we do, typically we will have the cells in culture for 4 hours. So one possible explanation for how TPO could result in monocyte production of cytokines is that it could be an indirect effect. In other words, TPO stimulating, say, progenitor cells, and then those then secreting something that acts on monocytes. So we wanted to address that potential issue. And so we sorted out CD14 positive monocytes, put them back into culture, and then looked at TPO stimulation. And what we found in this experiment from the same patients, sorted versus bulk, and then gated on CD14 positive monocytes, almost identical responses, indicating that, yes, that the monocytes do respond directly to TPO in production of these various cytokines. And so, obviously that would imply that MPL is expressed on these cells, and so we specifically stained for MPL on these cells. And what we found was that, yes, MPL is expressed, actually on a pretty decent subset of these cells, somewhat, again, to our surprise. And we then looked at MPL expression in these cells in conjunction with CD34. So here, these plots are showing MPL on the y-axis, CD14 on the x-axis for monocytes, and then color-coded for CD34. So here are your CD34 progenitors with high levels of CD34 and MPL expression. And then here, right here, sort of in yellow, these are your monocytes, which have high MPL expression, and they also maybe, to be fair, have sort of low but dim CD34 expression. So they're not necessarily, strictly speaking, CD34 negative, but more like CD34 low. So again, these might be a very unique subset of the monocytes that are the ones that are producing these cytokines abnormally, and it's something that we have yet to really delve into further. Okay, so to get back to more of the broader theme here, so amongst the different cytokines that we looked at by mass cytometry, we could, from these experiments, put them into essentially 3 categories. So the first 2 categories are cytokines that responded in some fashion to TPO, TLR ligands, and were either plus or minus sensitive to ruxolitinib. So of that group, the first category shown in green here was pretty sensitive to ruxolitinib. But the group in blue, which comprised TNF, IL-6, and IL-10, was largely insensitive to ruxolitinib. So maybe this is, particularly sort of bad group of cytokines. And then you have this whole host of other cytokines which were completely insensitive to TPO or to ruxolitinib, suggesting that maybe they're produced through other pathways. And probably the most notable in this category is TGF-beta, which is thought to potentially be an important player in that bone marrow fibrosis. So perhaps there are other pathways that need to be targeted to ameliorate the TGF-beta production. So, again, the next obvious step then is to say, okay, well, what are the other pathways that could be involved? And so we looked at a number of different targeted inhibitors. So Pevonedistat, which inhibits NFKB, and I'll come back to that in quite a bit of detail in just a second. Trametinib, which is a MEK inhibitor, VX745, which is a P38 inhibitor, and then JAK inhibitor 8. And so we looked at the effects of these different inhibitors, and we found that depending on the cytokine, that there were variable effects, but that overall, these different targeted inhibitors often had an ability to at least partially reduce cytokine production. So, for example, here you have TNF. In this particular patient, you've got 21% positive cells basally. Pevonedistat had kind of a moderate effect, trametinib a more significant effect, et cetera, and then it was a little bit different from one patient to another. And then just to summarize that, this is looking at 2 different patients. Blue would be essentially a positive or an inhibitory effect from the particular cytokine. So you can see in the case of MIP1 beta, there was a pretty significant inhibitory effect for most of these inhibitors. Some cases it was not really inhibited at all, in particular TGF-beta, again, not affected by any of these inhibitors. And then this is just another way of looking at data where we gated on the 90th percentile, so the cells that are producing the highest levels of cytokines, where, if anything, the effect is accentuated. Okay, so now I'm going to spend the next few minutes telling you about how we've specifically been attempting to target the NF-κB pathway and actually been able to move this into the clinic. Okay, so I mentioned Pevonedistat earlier. So this is a compound which is a NED8 activating enzyme inhibitor. And the way it works is that it acts upstream of the proteasome, and in a somewhat analogous fashion can result in this particular instance, in our example, IKB alpha, which is normally regulated by proteasomal degradation. Its levels accumulate, and that leads to a net negative effect on NF-κB signaling. So these compounds in this class are not identical proteasome inhibitors. They have overlapping but also distinct effects. And one of those, as shown here, is that it inhibits NF-κB activation. So how we got here is that after we identified NF-κB activation, obviously we're thinking about how can we target this pathway, and we looked at some various compounds that can target NF-κB, none of which are entirely specific for NF-κB, and after a fairly large number of experiments, we settled on Pevonedistat as being the most effective and also the most promising based on the experiments that I'm about to show you. So here is just confirmation that Pevonedistat does inhibit NF-κB in cells that harbor JAK2 V617F. So this is the HEL or HEL cell line that carries the JAK2 mutation. And what we found is that, for instance, when you stimulate with TNF, you get a decrease in, this is showing IKB alpha levels, a significant decrease in IKB, which is what you expect with shortly after stimulating with with TNF, you get a decrease in IKB levels, which leads to NF-κB activation. So that's what occurs normally with TNF. But if you co-incubate the TNF with increasing levels of Pevonedistat, that effect is completely blocked. So Pevonedistat prevents IKB from being degraded, which also then inhibits downstream NF-κB activation. We looked at proliferation of HEL cells in the presence of Pevonedistat, and so this graph shows you that Pevonedistat does have a dose-dependent inhibition of HEL cell growth, and here we looked at a plus minus ruxolitinib. So here is a dose response to ruxolitinib, and then these gray bars are different concentrations of Pevonedistat, and then adding in different concentrations of ruxolitinib. And so essentially what we found is that each of the two drugs had a dose-dependent effect, and that there was some degree of combinatorial effect when we had the two together. So we have also, in a preliminary manner, begun to look at the signaling effects of Pevonedistat in primary cells. So this is patients with myelofibrosis, looking at their CD34 cells, and these are our intracellular signaling molecules. And then we looked at basally, and then following exposure to TNF, Pevonedistat by itself, or the combination of Pevonedistat and TNF. And what we found is that, and this is in one particular patient, I'll show you another example in a second, phospho-P65 levels, they go up with TNF as expected, and that this was somewhat blocked by Pevonedistat. And the same in the inverse fashion was true of IKB, so more evidence that Pevonedistat is inhibiting NF-κB activation. We do see some effects on other signaling molecules, here phospho-4-EBP1, and a few others suggesting, and some of these can intersect with NF-κB, so we think this data does support that NF-κB signaling is the primary pathway that's affected by Pevonedistat, and so that's true. It seems to be the case in this patient, and somewhat similar effects in this patient as well. Now that's the signaling effects, so we wanted to go further and show that Pevonedistat affects NF-κB-dependent gene expression. And so we took sorted CD34 cells from patients with myelofibrosis, and then we did qPCR for NF-κB target genes following exposure to TNF, PEV, or the combination of the two. And what we found is that, as you would expect, TNF led to an increase in gene expression of these particular NF-κB target genes, and that this was inhibited by Pevonedistat. And so two different NF-κB target genes, we found similar results. So pretty good evidence that TNF is clearly blocking NF-κB-dependent gene expression. We've done similar experiments looking at the cytokine production, and if anything, have found even more impressive results with Pevonedistat. So here, this is looking at one patient compared to a normal and looking at IL-6 levels, and we see a strong induction with TPO, and this is almost completely blocked with Pevonedistat. This is the one you want to look at. And so very potently inhibits that cytokine secretion. And we also, in data I'm not going to show you, confirm that Pevonedistat does not directly inhibit JAK-STAT activation. So this really seems to be occurring through NF-κB. And similar results when we look at TNF. Here you see that ruxolitinib is not really having much of an impact on the TNF secretion, but Pevonedistat is much more effective. And this These experiments were conducted using 5 micromolar Rux, so a very high concentration of ruxolitinib, and 1 micromolar of Pevonedistat. And we just did another experiment, which I don't have the data to show you, but we went down to 40 nanomolar Pevonedistat, and it was still more effective than the ruxolitinib. So Pevonedistat, i.e. going after NFKB, was much more effective at blocking this cytokine production. Okay, and this is just another example with IL-6, similar results. Okay, so what is the effect of Pevonedistat on actual growth of these cells? So we've done colony assays using sorted CD34 cells from normal versus MF patients. And here the normal is shown in blue, the patient is shown in red, and we looked at increasing concentrations of Pevonedistat. So what you can see is that with increasing concentrations of Pevonedistat, there is an effect from Pevonedistat and a more robust effect compared to the effect of normal, which is obviously what you would like to see if you're thinking about using this as a therapeutic. You want to be able to ideally selectively target the malignant cells and not harm the normal. So yes, at higher concentrations, it does significantly inhibit the normal, but there seems to be potentially this window, at least in this colony assay, where Pevonedistat is more effective against the patient versus normal. We also looked at what the interaction is with TNF in the system. So TNF alone has a significant impact, and this is relatively small numbers, but relatively significant impact just by itself in actually reducing colony numbers. But I think the interesting point here is that when we add in Pevonedistat, if you look at the red, from here TNF by itself, and then TNF plus Pevonedistat, 0.2 micromolar, there's a more significant drop compared to TNF by itself, whereas the normal, that's not really the case. So it seems that perhaps there's this interaction between TNF and Pevonedistat, and this could be partly related to, if you think of TNF as being having pleiotropic effects, so it can stimulate proliferation, it can actually cause apoptosis. NFKB is more on the proliferation side, so if you block the downstream NFKB, i.e. proliferation angle, maybe the effects of TNF would be shunted towards apoptosis. We've also done this looking at the effects in combination with ruxolitinib, and again, with relatively small numbers, it seems that there is some degree of combinatorial effect with Pevonedistat and ruxolitinib. And that is very relevant because, as I'm about to show you, for the clinical trial that we have just recently initiated, we are combining Pevonedistat with ruxolitinib. And I wish I could say that that was because of we had this really incisive data. In fact, most of the data I showed you was generated after we had come up with this idea for the clinical trial, but really it was more of a practical matter, which is that these days most patients with myelofibrosis are treated with ruxolitinib. So if you want to conduct a clinical trial, it's going to be very difficult to find patients to enroll unless you allow patients who are already being treated with ruxolitinib. So that's what we did. So we designed our study to say, take a patient who's been treated with ruxolitinib and they just need to have stable disease. Essentially, you've achieved what you're going to get out of the ruxolitinib, which typically you see by about 3 months. Maybe you get a little bit more benefit for a couple more months down the road, but for the most part, you're going to get what you can get out of the ruxolitinib at that point. And so then we add in the Pevonedistat. So that's how we proposed it. And it's a dose escalation phase 1 study, so safety and tolerability is the focus. We are using very low doses of Pevonedistat to start, and then we'll dose escalate, and hopefully it'll be tolerated, and we'll see if we have any hints of efficacy. So that study, we just initiated that recently, and we just enrolled our first 3 patients, and they just received their first infusions starting last week. So we'll see what happens with that, but we're obviously excited about that study. Okay. So for the last few minutes of my talk, I'm going to switch gears a little bit. So virtually everything I showed you was focused on myelofibrosis and to some extent on secondary AML. This is a project where we focused on polycythemia vera. So we were intrigued by this paper that had come out in 2013 by Brady Stein and colleagues, and then I realized that Brandon McMahon is an author on this paper. I don't know if he's here, but one of your colleagues. And so it was quite interesting because in polycythemia vera and all the MPNs, it's well established that they're typically diagnosed with older age. But there is this unique cohort of patients, specifically with PV, who tend to be diagnosed at earlier age, and they have distinct clinical characteristics. So this is data kind of represented from their paper, where they specifically looked at patients who were 45 and younger at diagnosis, and then patients who are 65 and older at diagnosis. So this is not this is specifically a diagnosis. It's not patients who say were diagnosed at age 45 and then examined at age 70. This is when they were diagnosed. And some of the specific differences that you could see, the younger group was predominantly female, which was different from the older group. More splenomegaly, white count lower, JAK2 allele burden lower in the young group. And then there's this unique tie-in with splanchnic vein thrombosis in these younger patients. As far as the long-term outcome and transformation to MF or AML, these numbers were not significantly different, but I think the bigger point there is that the overall prevalence of transformation may not be different, but the rate is different. So in a younger patient, it takes longer to transform, and I think some of the data I'm going to show you may perhaps at least be consistent with that. Okay, so our question was, is there some difference at the genomic level in these two groups of patients? We know that by and large, every patient with polycythemia vera has the JAK2 V617F mutation. So are they otherwise different genetically, or is there some other explanation for why these two groups of patients seem to have distinct clinical characteristics? And we were reminded that it's been shown in part by my colleagues at WashU that acquisition of somatic mutations is a normal function of aging. And so, for instance, here is a study published by the group at WashU focusing on AML genomics, showing that there is a correlation between age and the number of somatic mutations in AML. And this is just showing, and we did a prior study in a patient with myelofibrosis who transformed to AML, and at least with those just two data points, we saw a similar kind of trend over time. And then this was looking at normal blood. Again, this study from my colleagues at WashU, they looked at individuals at different ages, and then they looked at individual clones at the number of mutations, and there was a strong correlation with age. And so we hypothesized that if this was the case, that aging was the predominant factor in mutation acquisition in PV, that we should see a similar type of trend. And so we decided to investigate that using exome sequencing. And so here is the cohort we assembled. It's not particularly easy to find these young patients, although, again, there's a unique cohort that exists. They're not that common. But we were able to find 10 patients who were diagnosed at age 45 or less, and where we were able to collect a sample before they had turned 45. So all the patients in our study were diagnosed, and then sample was acquired very shortly after they were diagnosed. So not much time had passed, which is an important potential confounding factor when you're doing this kind of analysis. So we got 10 patients who are younger, and then 9 patients who are older. And then subsequently, we expanded the cohort with additional about 10 patients in each group. They were not sequenced, but we used for other assays, including cytokine assays and clinical correlations. So our group somewhat resembled what I showed you with the Stein paper, in that there are more females, there's more a trend towards more splanchnic vein thrombosis, and again, young versus old. Okay, so we then did what we call enhanced exome sequencing, so traditional exome sequencing, but then we spike in additional probes for MPN-specific genes, and then we have this AML recurrently mutated gene set. So we add those probes in to get very deep coverage of the typical players, such as TET2, ASXL1, those kinds of things, and again, to ensure that we get good coverage of the known recurrent mutations in these diseases. So we did this work in conjunction with the McDonald Genome Institute at WashU, and we did have to remove a couple of patients from our analysis because there were some technical issues and maybe some sample contamination. So we ended up with, I think, 8 patients in the young group, 7 in the young and 7 in the young and then 8 in the old. And first, I'll just show you the JAK2 allele burden. So in the group that we sequenced, we could not show a statistical significance in terms of the difference in allele burdens, but we looked at a larger data set from WashU, from clinical testing, and we did find that to be the case. So maybe the sample size is just too low to really show that, but it does somewhat fit with the existing data of lower JAK2 allele burden in younger patients. When we looked at the overall number of confirmed somatic mutations by exome sequencing, we found that there was a significant difference in the young versus old. So in the young, there was an average of 3.4 mutations per patient, very similar to what we had kind of extrapolated from the prior publication I showed you. And so that would have predicted about 4 mutations. And the old group, we found an average of 10.4. So again, this is very similar to what would have been predicted. So this is all somatic exome mutations. We culled that list to what we thought were likely to be true driver mutations. So known mutations, again, in the typical players, ASXL1, TET2, et cetera. And when we use that kind of more restrictive criteria, we ended up with no other mutations in the young group other than JAK2. So in every single patient in our analysis, it was JAK2 and no other mutation that we felt was likely to be a true driver mutation. Whereas in the old group, every patient except for one, we were able to find at least one, and in some cases 2 others that we felt were likely to be driver mutations. So pretty stark difference there. And then this is probably pretty hard to read, but just to give you an idea, these are all the different mutations that we identified as being likely drivers. And so for instance, again, ASXL1, TET2, CHEK2, DNMT3A, et cetera. So, and we obviously had the different allele fractions for these different mutations. So then we asked in the old, because again, there were no other secondary mutations in the young. In the old patients, what's the clonal hierarchy? So you can infer to some extent just by looking at the numbers. So, for instance, in these you have, so JAK2 is in blue, and then these cases where the secondary mutation is in red, where it's a higher allele frequency, the assumption would be that that mutation occurred first, simply because it's a higher number. It doesn't prove it, but it suggests that. And so that's what the B is for before. And then if they're similar, you would, and I'll come back to this B versus C, but if they're similar, you would infer that they're probably acquired at a similar time and in the same clone. So that's the other question is, particularly if you have mutations with lower VAFs, are they actually in the same clone or are they in different clones? And this is an important point because, as I'm sure most of you are aware, there's been a growing understanding of this phenomenon of clonal hematopoiesis associated with aging. And so in these older patients, it could be that you could have a TET2 mutation with a 10% allele frequency, and it's actually not even in the same clone as your JAK2 mutation. You cannot distinguish that, for instance, in these cases where the allele burdens are relatively low, although by just the straight numbers, you would perhaps infer that this mutation occurred after JAK2. So we couldn't really make any firm conclusions, but I think the bigger point here is that, while on the one hand, I showed you that the total number of mutations is very consistent with what would have been predicted with normal aging. If that were the case, you might at least hypothesize that many of these secondary mutations in the older patients would be occurring prior to JAK2. So the idea would be, let's say you get a TET2 mutation, it's a CHIP mutation, it's just a clonal hematopoiesis, and then, so that doesn't give you an MPN. You get the JAK2 mutation later, and then you're diagnosed with polycythemia vera. That would make sense, but this data does not support that in most of these cases. So only in 3 of these patients did it look like, based on the numbers, the mutations were acquired before JAK2. So, long story short, we wanted to try to validate this by doing colony genotyping, and so we had samples available for this from only a handful of the patients, but we did, in those cases, attempt to do that, and so here you can see the distribution for each case. Here are the bulk VAFs, and then the presence of heterozygous or homozygous or wild type for these different mutations in each of these cases where we were able to do this. And so you can see how, based on these numbers, you can say, well, in this case, it appears that ASXL1 was first, and then JAK2 was second. Here it looks like they were acquired at the same time. Here also, at the same time, although JAK2 is homozygous. And this is a case where you have 2 different mutations in the same patient. In this example, we can't really tell if they're in fact even in the same clone, because we have low allele burdens and we have relatively small numbers of colonies that we could genotype. So while it's possible that ASXL1 was first, we just didn't have enough colonies to examine, it's also possible that they're actually independent clones. And then one kind of a really sort of unusual scenario is with this patient. So this patient was in our older group, and the data I showed you thus far was from this stage, where all the mutations had a similar allele frequency. However, we had serial samples from this patient who ultimately transformed to AML, and at the AML stage, the patient had some of the common mutations you see in more aggressive disease, RUNX1, ASXL1, NRAS, but what's missing here notably is JAK2. And so what happened is the JAK2 mutation was lost. And so that's this yellow clone here. it goes away, and then this clone evolves, which is the bad one, that gives rise to AML. But the confusing part is that ASXL1 is retained. So it was present here early, and it looked like it was in the same clone. But in fact, even though these numbers don't completely back it up, what we think was happening is these mutations were acquired a little bit later in a fraction of the cells, really the majority of the population here, but a fraction that was outcompeted by the clone that eventually acquired RUX1 and NRAS. So by doing the serial analysis, you can actually tease that out. So going back to this graph, you can see that, again, we have basically, these hashtags are denoting the ones where we confirmed what the actual clonal hierarchy is. And again, the take-home point is that there isn't one predominant pattern, and this is actually quite complicated. So ideally, you see a lot of clinical studies where they do this kind of analysis. They look at secondary mutations, but they can't really tell you the order or the hierarchy, and this is an important factor that could really be confounding these kinds of studies. Okay, so lastly, we looked at plasma cytokine levels in these patients. And going into this, I kind of hypothesized that maybe the reason why this particular cohort of patients who are young manifest with PV is because there's something wrong with their inflammatory milieu. And so they're more prone to, let's say you get a JAK2 mutation, that festering and then growing out. And in fact, based on our data with a limited number of patients, in fact, if anything, it seems that it's the opposite. So for the first point is that we are actually able to obtain age-matched controls. This is important because we know it's known in the literature that as just a function of aging, so again, hallmark here is don't get old, but with normal aging, inflammation increases. So we wanted to make sure we had age-matched controls for our two groups. So we did that, and what we found is that here on the left is the comparison of young PV versus young normal, and then the right is old PV versus old normal. And essentially the same themes are present, which is that multiple cytokines are abnormally produced in both the young and the old in comparison to the respective age-matched controls. But perhaps the more interesting is we then normalize this data so that it all could be compared to each other in terms of what's the relative overproduction of these cytokines in comparison to the age-matched normal. And what we found is that, if anything, it's exacerbated in the older patients. So the older patients have even more, to a more greater extent, abnormal cytokine production. And after the fact, I think if you think about it, it makes some sense perhaps that these patients have more mutations, and maybe the inflammation is contributing to whether or not it's the actual acquisition or the clonal outgrowth of these different mutations. Okay, so this is a very preliminary study and needs to be validated with a larger number of patients, but I think it raises a lot of interesting questions. So, first, no one has, this, as far as we know, really specifically looked at this before, so we think it's significant, even though, again, it's a relatively small sample size. But it does raise questions about what is really the fundamental requirement for disease initiation in PV. Again, we know that some of these patients have just JAK2, some of them have JAK2 plus other things, and what does that really tell you about the fundamental nature of this disease, and what is that actual interaction between aging and acquisition of secondary mutations? That little snippet of data I just showed you, to me, suggests that there might be a role for age-specific inflammatory cytokine production in modulating PV disease phenotype. And I guess that's it. So I'm going to wrap up there, and I want to acknowledge all the current members of my lab who've contributed to this work. Daniel Fisher is a postdoc and now staff scientist who's really driven a large part of this work. Former members of the lab as well who have been important contributors. My clinical research team, who's been really key, and then as we've translated this to the clinic, obviously are involved in that. The CyTOF work is in conjunction with our monitoring lab, which is part of our Center for Human Immunology and Immunotherapy program. So a number of staff there have been instrumental in that work, and the patients, of course, who have donated their samples for our research and are participating in our clinical trials, and then my funding sources. So thanks for your attention, and I'll be happy to take questions.
Last stuff you were talking about, and going back to the earlier stuff. So you showed that even when you could greatly reduce the immune burden, you still had the inflammatory phenotype of the bone marrow, monocyte producing and so forth. But how much of that was probably present? In other words, as you talk about, elderly people have more inflammatory cytokines, but it's also incredibly variable. You can have a 7-year-old that has an inflammatory profile of a 20-year-old, and but on average they're going to tend to be higher. How much of that increase was not so much due to the MPN, but due to the fact that the people most susceptible to MPN had the highest inflammatory cytokines.
Yeah, that's a good question. I mean, and Eric and I were kind of discussing stuff related to that as well earlier. I mean, one question just to relate to that we've wrestled with when we were doing this, putting the study together was, what is normal? So, you get what's an age-matched normal? An older person, do you take patients who have diabetes and hypertension, is that normal? Or do you only take healthy normal, right? So that's a big factor. In our study, I think we got it from a company where there was just like, whatever healthy volunteers had agreed. There's no specific criteria. So that could be a big factor in that variation. But, so it's really hard to tease out. But I would say in our data set, the difference between young and old was actually very minimal in the normals. So that implies that the bigger difference is from the patients themselves. Yeah.
So going back to mostly the earlier cytokine data, so you painted a pretty complicated picture of multiple cell types responding by expressing cytokines. And I wonder if you could say a little bit more about who exactly cell type is required, beyond those mutations to initiate or maintain the disease. Because I got a little bit lost.
Yeah, sure. I mean, you're not the only one, I'm sure. It's very confusing and complicated data at times, and I'd like to say that when you're working with these primary cells, it's messy, so it's never going to be like totally clear. But to answer your question, in some ways the simplest or most direct way to try to answer those questions is be in mouse models. So, for instance, in mouse models, we know there are a variety of JAK2 knock-in models. So first of all, JAK2 alone is sufficient to induce an MPN phenotype, most typically polycythemia vera. That raises questions as to, what do you need for myelofibrosis. But also, you can transplant the disease only with long-term HSCs in those models. Okay, so that's one point. But as I showed, we think monocytes are an important player. So that's something that it's kind of on our priority list. It's not number one, but one of the things that I would like to go after is what is the actual requirement or contribution of these monocytes in initiating or maintaining disease, and that's something, again, that I think most directly can be examined in mouse models. But nothing has been published in that regard as of yet.
Great. Thanks.
All right, thanks.